Okay, so hello, my name is John Roiser. I'm from University College London in the Institute of Cognitive Neuroscience. Let me thank the organisers for giving me the opportunity to present here. I think it's actually an incredibly worthwhile and valuable meeting. I was very motivated to come and speak here today, so thank you very much for that opportunity. And let me also say that if you miss something as I go through the talk, don't be shy, put your hand, that's fine. And maybe I'll try and explain a little bit better. And the chair, if I'm going over time, then cut me off, because we should try to keep the time a little bit.
Sorry. Could you please?
Oh, would you rather?
Somehow the microphone near you do this.
Okay, I'll tell you what, I'll sit.
Okay, thank you.
Okay, so what I'm going to be talking about today is what we actually measure in brain imaging and what kind of analysis we do on those measurements, and then what kind of conclusions or inferences that we are able to use from the statistics that we perform on our images. And as you can imagine, this is a highly complex process, and it can also vary quite a lot between different places. But some of the things that I'm going to be talking about are quite general, and then some of them are quite specific to one particular program, which is called Statistical Parametric Mapping, or SPM. But actually, in general, pretty much all brain imaging packages use somewhat similar techniques in order to achieve the goals of drawing conclusions. So most of what I say is going to be generalizable, though Professor Bullmore, I'm sure, will correct me with regards to other imaging software packages, one of which he invented. And one thing I think it's important to understand right from the beginning is what we're actually measuring. So there are a number of things that you could imagine that we might measure, and the first thing that you might naively possibly think that we could be measuring in brain imaging is the brain cell activity. But unfortunately, we're not measuring brain cell activity, so that's definitely not what we're not measuring. What we're trying to do is get a handle on what the brain cell activity would have been by measuring some kind of surrogate measure, so trying to understand other measures that probably correlate with that brain cell activity. And there are actually lots of different things that we can measure depending on the particular technology. So we could measure blood flow, for example, because different parts of the brain, when they are responding, use up energy, and that needs to be replenished, so we need to deliver glucose and oxygen to the parts of the brain that are working hard. So we can measure blood flow itself, or glucose metabolism, or we can measure blood oxygenation. And in fact, in most brain imaging studies, what you're probably going to be looking at measurements of is blood oxygenation, as we will see in a rather circuitous route. So let's talk about first the things that people don't use so much anymore, but at least we knew what we were really measuring. So that sounds like a slightly pessimistic statement, but unfortunately in modern brain imaging experiments, those who use functional MRI, what we're really measuring is not, doesn't have any units. So the thing I like about these old positron emission tomography studies, which of course are still carried out, but fMRI has superseded them a little bit, is that we actually get units for the things that we're measuring. So we're really measuring the actual amount of blood flow, so the milliliters of blood per milligram of tissue per second, for example. You get measurements out at the end that have some units like that. So the same with glucose utilization or glucose metabolism. So that was quite good, because that we could compare across different sites with not too much difficulty. However, PET studies have the relative disadvantage that they have not only quite poor spatial resolution, quite good there, but relative to fMRI, they're quite poor. So on the order of maybe 8 to 12 millimeters in the old studies, and in the new PET scanners maybe 4 millimeters. So you can get quite good images, but not quite as good as with MRI. But in particular, they have very poor temporal resolution. So you need to collect data over several minutes in order to get a good idea of the signal that you're trying to measure. And of course, in psychological studies, when we're trying to change the task demands over a short period of time, that presented us with some difficulties. So the kinds of experiments that you could do were limited by this ability to measure the signal quickly. One thing that brain imaging studies with PET really still have the advantage on is measuring the brain's biochemistry. So it's one thing to measure the brain activity, but I should also measure that, say that in terms of receptor binding, so if we want to understand how many dopamine receptors does a subject have, or what concentration are they, or how much dopamine gets released when we give a subject a particular stimulus, we can only use PET, so that's all we have. So there's a very beautiful set of images here which I rather like, looking at healthy volunteers who were either family positive or family negative for alcoholism, and you can see that even in subjects who aren't suffering from alcoholism themselves, there's a difference in the brain chemistry in this area called the caudate in terms of the dopamine system. So that's something we can still only really do with PET. But for the most part, modern studies of brain imaging and psychology really use magnetic resonance imaging, or MRI. And there are two forms of MRI that are commonly used. One is called structural MRI, and that gives us a nice clear picture of the brain structure, maybe down to a 1 millimeter voxel. So a voxel is a volumetric pixel, a pixel in 3 dimensions, so little boxes through the brain. So 1 by 1 by 1 millimeters, which is quite good, so you can see it very clearly. And you can tweak the MRI machine in order to produce different types of signals to allow you to pick up white matter or grey matter, depending on what you're particularly interested in, so whether you're picking up the cells themselves or the connections between them. And of course it has the value of having really good spatial resolution, and that's what MRI tends to be used for in hospitals most of the time, obviously. But the part that probably most of you are most interested in is the functional MRI. So we can analyse something related to brain activity, I put activity in inverted commas, while the participants perform a task. But of course we're not measuring brain activity itself. So the value of fMRI is that you can measure things over short time periods, so you can give stimuli a couple of seconds or a couple of maybe 5 or 10 seconds apart, as opposed to a couple of minutes apart, so that's very useful. But it does give us a very indirect measure. So what we're really looking at is something called the bulge response. That's the blood oxygen level dependent response. And the bulge response relies on some very simple physiology, which I hope you might remember from school, and that is that your red blood cells, you can see a picture of them in the top at the middle, carry this marvellous molecule hemoglobin, which attaches to oxygen and carries it around your body for you. And this particular molecule, this protein, has a different magnetic property depending on whether it happens to be bound to oxygen or whether it happens not to be bound to oxygen. And that's what MRI takes advantage of. It takes advantage of the fact that the oxygenated relative to deoxygenated hemoglobin provide different physics signals from the scan. So that's what MRI measures. It measures magnetic properties. So this gives a marvellous ability to pick up areas where the relative ratio of oxygenated relative to deoxygenated hemoglobin varies. And so that's what we're actually trying to measure when we're doing functional MRI studies normally. We're not measuring activity, we're not even measuring blood flow directly or glucose metabolism. It's quite correlated with blood flow, as you might expect, but it's not really anything that we can assign units to, which makes it a little bit more difficult to interpret sometimes. And so what we're really looking at